It's live now, by the way.
I'm glad that was the first thing on the stream.
So the bathroom is. Oh, halfway. Like closer to the. Yeah. And it's up. Okay, got it. All right. Just a quick audio check. Can y'all hear me? Nice. What? You're not. All right. Oh, look, Joe is here. I assume that's who Juicy J is. No, it's not? Oh, shit.
I think it's no, we can't hear you.
Just no.
Yeah, no means no.
Just know your enemy. Latvia. Jewel of the Baltic.
Wait, can they hear me?
Probably. Mike Tassia is also in the chat.
I'm on here.
All right, video should be working now. Yep. Thank you, Megan. And Mike, I will try to live up to your expectations. I'm aiming for just short of living up to everyone's expectations today. It is cut off. That's bizarre. I'm going to try and fix that video issue while we're here. Sometimes we do things in the name of rhyming and assonance that don't reflect our real feelings. Sponges aren't really outdated. Trichoplax is overrated, though. I'm just kidding. Trichoplax is great.
I'm on YouTube right now. If you don't want to enter the meeting, that's fine. Oh, it's upset down, please. Oh, this is your phone. Okay. That's like what?
All right, I think the video should be fixed now. I also promise that I don't actually say anything defamatory of sponges in the talk. Still not visible for the. What's not visible? Really?
It says live in 11 minutes now, even though it has. Oh wait, no, I just need to reload it.
Okay. All right, we got 12 people watching. I said if there were more than 6 people watching, I would consider the stream a success and worthwhile.
What if there are more people watching the stream than are here?
I think that'd be great.
So I have a question.
What's up?
It's about the title.
The title, yeah.
Singapores aren't killing me, right?
It's very interesting that you should say that. You'll see in a second. I'm making use of a rhetorical device. Or not really a rhetorical device, but I'm using it as an opportunity to grandstand on something. What is rhetoric? Oh my god. I'm gonna have to like do my best to not look at chat.
Can you minimize it?
I could, but I'm not going to.
Yeah, there might be legitimate questions on that. You know.
My God, we have electric lights here, people.
All right.
We're a modern society. Hey, how's it going?
Pretty good.
Yeah. Okay.
I'm excited. I'm keyed up.
Okay.
I'm set up. I'm streaming.
You're streaming. Good. Over in the air?
Yeah.
Yeah, okay.
It's going out to my YouTube channel.
Right. And your family or somebody, or some people that you know
Yeah.
can see it? Yeah, because I'm
Colleagues.
not sure about the speed. Hmm? Are you offering to bring me a coffee back?
Yeah.
That would be awesome.
Black.
Thank you. Yeah, that's great. Thanks so much.
Yeah, my parents weren't able to make it, so
Yeah.
they will be watching.
Okay. But it'll be recorded also, right?
Yeah, there's going to be an archived version on my channel as well.
So, yeah, okay. Where are your family?
They're in the Dallas area.
Dallas. Okay, yeah. Okay, great. Good. Yeah, we had people doing this before. I just hope the connections, keeps up. But probably, now it's summer, there's just much less traffic on the campus. With the
Yeah.
students and that stuff, so,
The signal quality has been good. Yeah, it should be pretty neat.
I could check by, internally. I just go
Oh,
to
really?
the YouTube thing and see.
Yeah, I can send you the link. I think actually all these 3 in the back are friends of mine from college, and I think they're all on my YouTube channel, spamming the live chat with.
Okay. Posting
That's a joke.
derogatory comments.
Mostly helpful. Mostly helpful.
Okay.
Only a very small amount of trolling, which I'm taking as a.
Well, what do I just search for? Townsend?
If you search for my name, my channel might come out. If you also search for the title of the talk, it will come out, or like James Townsend's. If you search for
Tina for no one else is going to be streaming on that.
if you search for I think I title Tentacular Spectacular Jellyfishertation, it should come up.
spell that. What is it? Tentacular? Tentacular?
Tentacular?
Spectacular.
Yeah. I think I'm also just gonna.
You'd be amazed at what. You'd be amazed at what actually pops up there.
Oh God. I should have thought that through.
Yeah.
Oh shit. It's League of Legends.
Yeah, it's like what? I'm looking. It's feel like I'm looking at my son's computer.
I just sent. I sent you a link.
Okay.
Well, I wanted to title the species description manuscript "What's black and orange and red all over?" This ctenophore we found,
Oh, yeah.
but you know.
Got it. Nice.
Everybody, my committee chair is in the live stream now. Joe Jordan says hi from Latvia.
Latvia?
He's in Latvia for some reason. No, it's for his wedding anniversary.
Vacation.
It's for his wedding anniversary.
Wedding anniversary? He married a Latvian?
I don't
Well,
think.
It's got nothing to do with Latvia. He just likes to travel.
Yeah, exactly. He's in Sweden for work.
Wow. So is somebody here, Victor?
Oh, that's one of my friends from high school.
And
Damien.
Michael?
Damien is from Auburn. Mike's actually one of my co-authors.
Yeah. I'll just get off so I don't consume bandwidth.
I like the idea of
Actually, what I really need to do is pull up the thesis.
I think it hasn't been streaming using a lot of pins, but yeah, because it's the same data and it's the yes, right. But I guess even like in that case, it looks pretty good except for sound. I'm gonna Katie is expressing her deepest sorrow. She didn't get caught. She didn't get caught.
That's okay.
Hey, Paul. Paul Janvey, something.
Yeah.
I know I'm unfamiliar with something you're saying. Yeah, he's like the editor of Soft Matter. He sent me manuscript to read it, and I just got an email saying it's overdue.
He sent you a manuscript for what?
He's an editor, so of Soft Matter.
Oh,
it's a
okay.
journal, so he's sending that to you. That's what your friends do. Like,
Okay.
they spam you away. So a thankless task being an editor, you've got to get good, find reviewers for manuscripts.
And no one wants to grease the wheels of.
No.
Wow, we'll be busy, you know. So this is a new journal.
Cool. I hear good things.
Yeah, the problem is, like, you know, the more journals, the more editors get notes into you, the more requests you get.
Good luck. Don't get lost. Do you guys want my key card? Take my key card. Take my key card.
All right. Yeah. Which one of us is most likely? Dragon.
Thank you. That's so kind. You know me, my coffee. Okay.
That ellipsis hides a great and dark secret.
Yeah, they closed it too, so yeah, it's the best coffee.
It's obviously the best.
Thanks. When you're actually talking, I think we'll turn off this one. Maybe we have one in the back, or maybe we have it off. I don't know. It's getting pretty noisy.
Okay.
Fine.
Thank you. I brought it from home. Oh my god. Yeah, it's just a little better quality than the old webcam microphone, which are terrible. It's true. How are you doing? VTA or something?
Oh no, we're having our third year meeting. Where Kim talks to us for half an hour and we have free food.
Cool.
This is great.
Yeah, for sure. Free food's legit.
Yeah, and then I could have gone back to that meeting if I pressed it, but my guy just came back from vacation, so I'd rather not. Because he's just gonna ask me what I've been doing, which is nothing.
Yeah, but that's the great secret of grad school is everyone's somehow doing that.
I mean, I said to him before because I actually did take one of his vacations and there's nothing else to take him on, so I'm like talking to him back. And I don't want to deal with it.
I heard that. so nerve-wracking, dude.
You're gonna rock it.
One of the wisest things I heard recently was I was talking to this guy that I sort of talking to a lot of advice with Townsend, and he was just like so nervous that that's going to go bad. He's like, what happens if that's going to go bad? Oh, well, who's it then is that your ideal emotional or physical state?
That's a good point.
He's just like, don't get around by bus. If you're taught. And I was like,
That's weird.
yeah, he had a lot of funny stories. Is that KK I hear? Do you go by that?
What?
Oh no, that's not. Is that? Yes, no, that's not Kelly Karch. It's Hannah Richter. Oh, thanks, Ken. Or thanks at Ken. You made it.
Did you bring any snacks?
There are snacks inbound.
There's Indian
There's
food.
Indian food left over.
Did you
from
bring?
us.
Did you bring food with gluten only?
No. I think there's grapes coming.
Do they have botrytis?
No. Maybe. I hope that's not a trick question.
I have a friend. This is complete lie. He lives in a farm. He lives on a farm. His table has some kind of shrine. Can you clean this, please?
I can lick it off, but that's all I can promise. Now Kelly Karch is here.
Hey,
Are you waiting for me or something?
Joe was asking.
Is there a bathroom?
Yes, that we do.
Where? I know his lab, but where else?
down the hall.
Just started.
On the left.
Can we just say you're done?
Okay, I'm down.
All right. How are you?
Oh, nice.
Paul is an editor of Soft Matter.
No, you said
Man
it
is
around
like
for you back again.
insight, and I'm overdoing it. It's
Just tell me before we see it.
That's what happens. Like, you get people, you get to know people, and that's what your friends do. They stick enough journal on you. Like, I've never used Soft Matter before. It's like, now I'm not an editor.
I got my favorite colors. Yeah, then you leave the front for the stare at that. Yeah.
Hey, what's your problem?
Oh, thank you. I was like,
What's your Virgil's trying accurate idea.
That was me. Well, we have food here on Friday afternoons, so actually that Friday. That's what, man, that's what the chapter told us. It's still pretty good.
Still, there was like visible.
Yeah, I know.
Okay, I'd like to point out there's no word that has CT as the first two letters.
huh, weird.
How do you pronounce it?
We have a whole primer,
How do
but how posh do you consider yourself?
how do you pronounce it?
Tenaphore.
Tinaphore?
Or
Only
tenaphore.
question I had, I was saving it for the end.
Or
Why
tenaf,
C?
because it's Greek.
And this is America.
Yeah.
This is America, so we accept Greek culture. We cherish and celebrate Greek culture.
I'm done with my question.
Nature.
This is the Wisconsin. This is one of the great architectural marvels of the
Oh, yeah. That's what architects call the learning building. So he didn't get it right. Like the Salk Institute, which is an experimental project.
Yeah, Britain Chance looks just like ours.
Oh, yeah.
He used to be out of my
Just like that other guy, John Nash.
Oh, yeah. But
All right, should we get started?
It's not up to you.
Okay. But should we?
No, I don't think it's up to you.
No, it's not.
Okay.
Just
Yeah,
say
we'll
all
call,
first step.
we'll call on you when this is ready.
Okay, so this is Spencer James Townsend, his advisor, Professor Alison Sweeney will give a little introduction, and there'll be a public talk in which afterwards we'll questions, and then everybody will leave except the committee, and we'll squeeze into our next speaker. So, Alison.
Give it up for Allison.
I'm Allison. This is James. James is a sixth-year student in my group. He joined my group initially to work on structure of reflectin proteins, but on our sort of initial forays into the marine realm, we discovered taxon ctenophores, which is going to tell you about, and sort of fell in love with them and took it and ran with it, and have worked on several aspects of ctenophore biochemistry and physiology in his time in graduate school, and I believe he's going to tell you about sort of three topics. So ctenophores are interesting for a number of reasons he will tell you about, and very little is known about them, and so James has done a lot of exploring of the basic biology of these animals in the last 6 years.
Thanks for that introduction, and thanks everyone for being here. Hopefully some of you at least saw, switch to that window. There we go. Hopefully some of you saw my title. I'm going to make good on my official title in the poster. So I just wanted to lead off, someone asked me earlier, aren't ctenophores not jellyfish? In a sense, yeah, it's a terrible word and no one should use it. So unless you're trying to make a point and stand on a soapbox in the middle of your thesis defense. The reason why it's not that great of a word is because usually when people say it, they are thinking of a very specific thing. They're thinking of a cnidarian with like a medusa form, and in addition to being not very inclusive, it's also got a lot of baggage, a lot of negative associations people have with jellyfish.
What's that thing on the bottom?
That thing? That's my phone. And below that is some kelp.
Waterproof phone?
I'm above the water.
Oh, okay.
But anywho, yeah, so the term has a lot of baggage, and in addition to being not very inclusive, it turns people off, I think. I prefer the term gelata, which Stephen Haddock at Mbari has suggested, because there's actually a lot of animals living the gelatinous life in our oceans. There's gelatinous polychaetes, gelatinous mollusks, things like salps. And so, jellyfish doesn't really touch on that. And also gelata sounds like gelato, so I think people like it better. Salps?
What word do you use?
Salp. Urochordates. Very intriguing animals. Mike in the YouTube chat knows a lot about them. Shout out. But today I'm going to be focusing on a particular phylum of gelatinous organisms that not a lot of people know much about, ctenophores. So this is a ctenophore, pronounced ctenophore, or ctenophore if you're posh, and know more Greek. This is Pleurobrachia bachii. I used it in a lot of my studies because it's very common in the Pacific Northwest, and very amenable to various things that I've done to explore ctenophore physiology. Straight away, I just wanted to give you a primer on their physiology, how can you distinguish a ctenophore if you were in a dark alley corner. The big feature for the phylum is these 8 rows of ciliated combs. That's why the phylum has that name. They use these little things like paddles. They locomote and reorient using them. And it's the characteristic feature of the phylum. And if you look really closely at them, they have this structure that makes them look like kind of fused together eyelashes. So they're not actually producing that light in the picture. They're just diffracting that light from a light source that I have on them.
What's that length scale there?
That is about a fractional millimeter. I think this one's
Tell it to me in the size of that human hair. No, I mean yeah. How big is that?
It's a little bit less than a millimeter.
From where to where?
From here to here. Yeah. And there are bigger and smaller comb rows, but it's very interesting because these are the largest animals to locomote by ciliary action, which is not wildly efficient, but they make it work. The other very common but not necessarily universal feature of ctenophores are these tentacles. And it's, cnidarian jellyfish have tentacles, but those sting. These tentacles are different. They don't sting. We're going to touch on that in just a second. They're actually just sticky. And they have like a very interesting way of sort of fanning them out that they use to catch their prey. So why should you care about ctenophores at all? Probably haven't cared about them until this talk. Hopefully you will after. But ctenophores are very interesting for a number of reasons. Probably the hottest reason right now is because they're in the center of this big evolutionary biology debate about the branching order of different animal taxa. Basically, like one of the central questions that people harp on a lot in EvoBio is like, what was the first extant group of animals to branch off from the rest? And for a long time, people said it was sponges based on like morphology and early molecular data. But as we've added more taxa to molecular analyses like sequence data, some people, more and more people are getting this result where ctenophores are actually the first taxon that still exists to have branched off for the rest. And it's really bizarre and not intuitive because ctenophores are perceived as being complex. And it's kind of upset the apple cart in a lot of ways. But one of the things that's really missing from this debate that mostly centers at the moment on like methods of like analyzing giant data sets of sequences is actual like detailed knowledge of the physiology of basically any of these animals. We're focused in this debate on cnidarians, sponges, and ctenophores. We don't actually know all that much about a lot of their physiology, in particular like the more unique aspects of their physiology, because people just haven't really gotten around to studying them. So I've spent a lot of time working on things in particular about ctenophores that are unique to ctenophores, because I think that's like a very good lens to study and understand them through. Okay, so for this first part, I'm going to be talking about a particularly unique and enigmatic ctenophore thing. I'm going to be talking about why those tentacles are so sticky. So I said that they don't sting. They catch prey by adhering to it like fly paper. The tentacles actually like release an adhesive on contact with the prey. And the cells that are responsible for releasing that adhesive are totally unique to the phylum. And as a result, haven't really been studied very much and are still pretty poorly understood. They're called colloblasts. So there's no obvious homologs to colloblasts anywhere else in the world of animals. They're kind of vaguely similar to cnidarian nematocysts, the stinging cells in that group, but it's kind of like a superficial comparison ultimately. They have this really weird physiology. It looks like maybe like a dandelion or like a bouquet of flowers, and it's tethered to the base, to the tentacle itself by this like stalk. And the tentacles are typically covered in these cells. These granules on the top contain some kind of proteinaceous adhesive. We don't know really anything about the adhesive beyond that. The last major study of this was done in the late 70s. So it's like an interesting jumping off point because it would be neat to know, like, is this adhesive itself homologous to other marine adhesives, even though the cells themselves are not? Is it an interesting biomaterial in other ways?
How big is that?
These are like 1 micron. Because I pulled this from someone's like very old manuscript.
I see.
Yeah. So there's like a bunch of features.
They're pointing them out.
Yeah. There's a variety of interesting things about colloblasts, but I'm just focusing on this particular aspect of the physiology. Okay, so starting from basically nothing, we asked ourselves, like, okay, what are some possible mechanisms of action for this adhesive? And one pretty reasonable starting point is to ask, like, is it like catecholic in nature? So catechols are just this little chemical group that I've circled here. It's just like a benzene ring with 2 ortho-positioned hydroxyl groups. And it is found in like a ton of different marine adhesives. It's found in mussels, the little holdfasts that they attach to rocks with. make extensive use of this chemistry. It's found in polychaetes that live in tubes. It's found in squid beaks, all over the place. So it appears to be like relatively widespread. It could just be because the chemistry is really easy to do. So it's an interesting place to start looking. And conveniently, there is a 3-part colorimetric assay for detecting catechol called our nose reagent. So basically, our nose reagent, you apply it to a tissue or a solution sample, and if there's catechols in the tissue, it will turn red or orange. And a lot of times it's said to be like a specific stain for DOPA, but it actually just stains any catechol pretty promiscuously. So pretty easy to do. We get some ctenophores. This is one that I have fixed in formaldehyde and bisected, and I treat it with our nose reagent, and the tentacles turn orangish red. So this is a positive result for this stain, which suggests strongly that there are catechols present in this tissue, but it doesn't really tell us much specific beyond that. So we wanted to get more spatial resolution for this and maybe start to see like, what we're seeing localized to any particular structure on the tentacles. So we made use of this really neat old school technique called formaldehyde-induced fluorescence. And basically, if you apply formaldehyde to a tissue, the way that fixation works is it just makes random crosslinks. And sometimes random cross-linking will cause otherwise not fluorescent molecules to become fluorescent or to change their fluorescence. So conveniently, people have figured out how to do this to distinguish like dopamine and serotonin from other molecules in like nerves. And dopamine, it actually works because it basically has a structure similar to this, but with a catechol moiety on it, and it forms a fluorophore. And so people have done a lot of work on this in rats, and there's this interesting thing where it's even a little bit diagnostic. If you have like more substituents on the ring, then it'll like redshift the peak emission. And you know, it's pretty obvious when you see it, like it's green, if it's like a DOPA-like thing or a catecholic sort of thing. Serotonin-like molecules are more like yellow. So we low and slow fixed a bunch of ctenophore tissue in paraformaldehyde and put it on a confocal that had a detector with like a spectrophotometer. So the emitted light from this is binned into 3.2 nanometer bandwidth bins. And what I'm going to show you is like a true color representation of those bins where it's been translated into kind of like the appropriate color of light. So first I image the tentacles themselves. And lo and behold, the actual granules on the exterior surface of these tentacles fluoresce green under UV light. The whole peak is, or the whole emission profile of the granules themselves is redshifted relative to the tissue background. The tissue background is always going to be there. If you've done any like confocal microscopy, you probably run into this. Just all tissue has kind of a hazy blue background. So this is pretty distinct in relative to that. And you know, it has this mixture of different peaks, which actually kind of corresponds with what we might expect for like a multiply substituted population of fluorophores, because there's also concentration-dependent effects and stuff. So, very encouraging. We wanted to do like sort of an internal tissue control. Hello. We wanted to do an internal tissue control. So we dissected out a portion of the comb row. And we did this because there's no calloblasts in the comb rows. And also, ctenophores are very well known at this point to not use like catecholamine neurotransmitters, so they don't have or produce or use dopamine, epinephrine, norepinephrine, anything like that. So we wouldn't expect to like randomly find like dopaminergic neurons in here. But we actually saw these little green granules, not at the same frequency that we see them in the coloblast where they're everywhere, but they're kind of smattered around against this sort of meshy blue background. So that was really interesting. This spectrum is a little bit different, but it still has this peak at 502 nanometers, and it still is redshifted relative to the tissue background. So like, what's going on there? How unusual is this? Actually, weirdly, people have observed something similar in sea anemones, in their tentacles and the nerves inside of the tentacles. And nobody knows what those granules are doing there, but people have done a little bit of more biochemistry workup and found that in those tissues it was a mix of like DOPA and DOPA with like more substituents added onto it, sort of like what I'm suggesting. But it's just sort of replacing one mystery with another, but it's still very interesting, and maybe suggests that there's some parallels between these different early diverging phyla. So the thing is, like, in the nerves, at least in ctenophores, yeah, they don't make dopamine. They don't use like dopamine receptors. They actually only have glutamate receptors. It's the only canonical thing that they have. But they're also predicted to use like a ton of different neuropeptides or hormones that have been identified but never studied experimentally. And these wouldn't really be detected by traditional pharmacological assays because they're not small molecules. And we don't really know anything about them. And a lot of them seem to be unique to ctenophores. So there's a huge possibility that the things that we're detecting in the comb rows could actually be associated with nerves and could actually be part of these like neuropeptides that nobody knows anything about. So to get at all of this, including like whether the things that we're seeing in those granules are proteins or that could be part of an adhesive as well, we actually need to demonstrate that what we're seeing is not a small molecule. It's present in or associated with a protein. So we took whole tissue homogenates, precipitated out the protein, and tested that. So here I've solubilized and precipitated out a bunch of protein and a very particular buffer suited for this. So you can see these are actually little white pellets. They might be hard to see in the back, I don't know. I treated them with our nose reagent, and because we've actually enriched the proteins here in this relative to the tissue, I think the effect is a little bit more pronounced and easy to see. These samples from comb rows and these samples from tentacles turn bright red-orange, whereas a sample from just like chicken egg white albumin that we wouldn't expect to have catechols in any form in it does not change colors. So it does seem like we can extract and enrich these catecholic-containing molecules, and it does seem like they're associated with at least proteins. So just as a check, I also did formaldehyde-induced fluorescence measurements on these to see if we had actually enriched for the things that we were seeing in those micrographs. So for the tentacle extracts, it does seem like we've mostly gotten, we might have lost some stuff along the way. We certainly seem to have co-precipitated some kind of like maybe peripherin-containing thing along with this, but in the diagnostic range of the formaldehyde-induced fluorescence assay, we appear to have enriched for what we were looking at in the whole tissue micrographs. And in these body samples, and I should note that when I say bodies, I've removed the tentacles and everything related to the tentacles from the bodies when I grind them up, so it should mostly just be comb rows and stuff like that. We seem to have enriched precisely the granules that we were seeing in the micrographs. The spectra almost overlap. And also, just as a check, the chicken egg white albumin is, approximately the same as the background, which is just generalized tissue background. Okay, so to finish off this part, I just did some immunohistochemistry. We had an anti-DOPA antibody that I applied to a bunch of these samples. So I'm going to show you the actual experimental and a couple of controls. So initially, this looked pretty good, like a good stain on the tentacles. We applied this. We can see these punctate structures that we were seeing earlier and that we've seen in micrographs. But it's a little bit complicated because the tentacles themselves become sticky when you fix them. So they actually bind non-specifically just to the reporter antibody. And the other control is a little bit muddy as well. So this is maybe not necessarily like the clearest thing, but this second control is an antigen specificity control. So the primary antibody should have DOPA on it. And it does change the way that that antibody seems to interact with the region of interest. So it's possible that there's some kind of interaction going on between the DOPA that's bound to the antibody and whatever epitope is present in the tissue, but that's a little bit harder to assess. We also did it for the comb rows, and this was a little bit more of a straightforward stain. We applied it to this tissue, and we actually see this hexagonal, like lattice type structure come out with little punctae. So it appears to have the same geometry as the subepithelial nerve net, almost exactly, like right down to the size of the hexagonal lattice parts. So that was really interesting. It seems like the antibody might be more sensitive to like less concentrated epitopes in other parts of the nerve net. And when we apply our control stains, there's no specific staining of that structure. So the antibody appears to work for this tissue, and it seems to be a little bit more amenable to immunohistochemistry. So the final thing that I want to talk about in this portion is like, how could ctenophores be making catechol like DOPA or things that are similar to DOPA if they're not even producing they're not producing like dopamine or anything similar. So what a lot of people don't necessarily know, but has always been out there, is Chlorobrachia and other ctenophores express tyrosinases, which is a totally different class of enzymes. Tyrosine hydroxylase makes the precursors for dopamine. Tyrosinase is better known as producing the precursors for melanin, as well as a variety of other molecules. So this being in the transcriptome certainly implies they have the ability to express the precursors for any number of DOPA-like molecules. And importantly, these tyrosinase core domains preserve the 6 histidine residues necessary to coordinate copper for the enzyme to be active. And we actually did some homology modeling of this, and the geometry of the binding site is also preserved. So this being expressed, if it takes this folded structure, should certainly be active, but just its activity is not enough to determine what it's producing, because tyrosinases are super weird, and just slight differences in the geometry of the binding pocket result in totally different products. But it's at least possible for it to be done using this protein. So yeah, just to summarize, we seem to have detected evidence of catechols in 2 unsuspected places in ctenophores, in the tentacles adhesive granules, and associated with the nerve net. For the tentacles, it's a little bit more straightforward of an explanation. It could be involved in the adhesive protein. But in the granules, we need to look at it a little bit more. It'd be worth trying to figure out, are these granules like filled with neuropeptides? Or is this involved in signaling? There's a lot of open questions here to work from. And also interestingly, this actually all sort of made sense in the end, because I was talking to Leslie Buponis at the Whitney Labs in Florida, and found out that they have evidence suggesting that colloblasts and ctenophore nerve cells are actually derived from the same stem cell tissue. So when I told her about this, she was like, oh, that makes sense. So they probably have a pretty similar enzymatic toolkit going on as well. Okay, next. I wanted to show you guys an investigation of the mechanical properties of this adhesive, which is also something that's never been done, that I did with an RU from the University of Puerto Rico, Gabriel. So just a quick rundown of what we did. We have this instrument that we call a poker. It has a probe that we lowered through a droplet of water, beneath which there is a tentacle on a glass slide. And we just poke the tentacle. We let it come to rest on the tentacle. It adheres, and then we pull it off. And we're measuring the force as we're pulling it off. And we look for how much force it takes to detach. And that's what we call the adhesive strength of the tentacle. So this is just like what an actual curve that we looked at would look like. And we're measuring and reporting this bit, the difference between like the baseline when it force on the probe when it touches the tentacle versus the peak amount of force that it takes to lift off. So there are a couple of things that we did to just start. No one has actually conclusively demonstrated that these granules are destroyed or pop when they bind to prey, or just the explanations have been like pretty coarse. So we actually demonstrated that colloblasts are exhausted as you poke the same spot over and over again. We either poke the same spot without cleaning the probe between runs, or with cleaning the probe between runs. And if you clean the probe between runs, you gradually deplete the amount of adhesive present. If you don't, you gradually get all of it out and it gets stickier and stickier. And so this was the first demonstration of that. We also used this as justification to say that for our purposes, the baseline force we set as 200 micronewtons on our probe. And if we had recorded a force value after liftoff of less than 100 micronewtons, we said that the probe hadn't properly adhered. And we report those numbers, but we don't actually factor them into our summary statistics. So next thing we looked at was we wanted to see how fast adhesion occurs. Because of limitations on the instrument, it's actually almost impossible to get this to go faster than 5 seconds, we have this curing time. And so the fastest we could get it on and lift it off was 5 seconds. Adhesion certainly occurs faster than 5 seconds, it appears. No word on it, how long it takes actually, but job done. We also looked at ambient pH and the effect of that on adhesive strength, because if there was DOPA in this adhesive, DOPA is pretty sensitive to pH. Its properties can be modulated by ambient pH and R and things like mussels. It does not appear to occur here, and there's a number of reasons that could be happening, just because the pH within the actual granules themselves might be very tightly controlled. There might not be time between adhesion and full curing to actually have the ambient water take an effect. But it was good to know. So all of this is in some ways a little bit underwhelming, or it was at the time. We were okay, so the adhesive works fast, appears resistant to ambient pH, and as we expected, it's depleted as the granules are destroyed. But I wanted to put it into a broader context of other organisms that have been studied. So I went through the literature and converted basically all the values for adhesive force in other systems that I could find into comparable units to ours. So I didn't say that I did this, but ctenophore colloblasts, if we take the average, if we say the typical ctenophore colloblast has an adhesive strength of about 200 micronewtons across a probe that's 0.5 millimeter in diameter, we get about 1,000 newtons per square meter of adhesive force, which is pretty weak, as it turns out, compared to other things. It's about the same order of magnitude, though, as orb weaver spiderweb viscous capture threads, which is really interesting because orb weavers and ctenophores have been compared really superficially for a long time, mostly just because people think of them as both being ambush predators. Obviously, spiderweb is different from an organ that's attached to an animal's body, but this has been something that people have thought for a while. So we're oh, well, maybe there's more mechanically to this comparison than initially meets the eye. So general question emerges, why would you have a relatively weak adhesive compared to other things? Why not just make it?
But the spider catches things in the air, right?
Sometimes, or it can be pretty close to the ground.
No, fine.
Oh yeah, air versus water.
Yeah. So the spider web has to deal with inertia.
Yeah.
And ctenophore doesn't.
Yeah. That's true, for sure. And also, like, a lot of ctenophore prey tends to be smaller. But sometimes spiders don't deal with inertia. They actually collect and harvest and eat pollen, weirdly. Some orb weavers actually get a lot of their nutritional needs through that. But yeah, some of it probably has to do with air versus water, for sure. But for ctenophores specifically, why would you want a weak adhesive? Is there any reason why this might be advantageous? So came up with a model. I want you to consider a hypothetical prey item. It has a radius, it has a density, it's some kind of copepod, so it's about the same density as water. This value is pulled for the literature for copepods. But because we have a density and we have a radius, we can calculate a volume, we can get a mass. I want you to imagine that a tentacle adheres to the prey. The tentacle has a width. We actually measured this from our micrographs. It's about 50 microns. And say the tentacle adheres to the surface of the sphere and wraps about a third of the way around. So we can calculate an area of contact. And then, you know, just for the sake of realism, let's say that multiple tentacles can and probably do adhere to a single prey item because they're really, really close together. It's somewhat inevitable. And we'll say that n tentacles can adhere. Okay, so then our adhere prey will try to escape, and it'll escape with a force. Conveniently, someone actually has filmed a bunch of copepods and hydrodynamically modeled the peak escape force. So they've computed this like mass-dependent escape force, which actually works out to have the same units as acceleration conveniently. So all copepods basically have a mass-dependent escape force that they can generate of about 100 newtons per kilogram. And it has to do basically with like their physiology. That's the upper limit. So we have an acceleration, we have a mass, we can get a force. And then let's say that the force of adhesion is directly opposed to this escaping force. And we already said it was about 1,000 newtons per metered square. And that's going to be distributed over our area of contact, which we calculated, multiplied by the number of tentacles that actually adhered. Let's express this as just a summary value. So in this case, we're taking the difference between these things. If the number here is positive for a given set of dependent variables, then adhesion is the greater force, and the animal will stay attached. If escape is greater, the number will be negative, and the animal will detach. So we compute this out. We get this very interesting sort of manifold where like really, really small prey under like 0.5 millimeter in radius, pretty easily and reliably adhere most of the time. Tentacles attach to them. But as you get bigger and bigger, there's kind of this filtering effect where you need more and more tentacles to, or tentillae to be, attached before you can reliably catch the prey. And past a certain point, up to like 3 millimeters prey size, a 6 centimeter or a 6 millimeter diameter prey, it's really, really hard, almost impossible for the animal to conceivably handle it. And that actually makes a lot of sense because most pleurobrachia are about 1 centimeter in diameter, so that would be much bigger than the animal could even fit in its mouth. And if you just change the adhesive pressure, adhesive force by a factor of 10 in each direction, just to illustrate, you totally change the filtering behavior of the system. If you make it a lot weaker, almost nothing can ever adhere, which certainly doesn't happen. If you make it a lot stronger, basically anything will adhere all the time to this animal, even things that are wildly too big and would probably hurt the animal if they tried to actually escape. So if this is accurate, it seems like it gives the animal an ability to filter by size without actually being able to see anything. So that's just one possible justification for all of this. We don't necessarily know how much force it takes to hurt the animal, to detach a tentacle or cause damage. But this is just one possible reason this could be happening. The adhesive force is probably different on different surface chemistries, but the model still probably applies reasonably, just changing that adhesive pressure mechanism or parameter. And yeah, it seems like there is sort of an analogy to spiderwebs. Spiderwebs actually also filter things based on like the mesh size of the web, the position, height of the web. So there's a lot going on there. There also might be more things going on with ctenophores. They can actually change the spacing between the smaller secondary tentilla, and they can change the number of tentilla on a tentacle, or like different ctenophores have different numbers of them. So there's probably a lot more going on there as well, but it'd be interesting to do some more figuring on that. Okay, and now I'm going to do something wildly different, so you can wake up. Yeah, ask me.
One, what's a copepod? And two, how does it generate the force?
A copepod is like a little baby shrimper, little baby amphipod or crabby thing.
So it doesn't have a shell.
Well, it has like a chitinous exoskeleton.
Okay, but it's not like a shellfish. It's like a lobster.
More like a lobster, less like a clam.
And so that F net is coming from it like
Paddling desperately away.
paddling.
Yeah. Flailing.
Yeah.
Okay. Did you have another question?
No, that was the question.
Okay. What's up?
How do they transfer the fluid from the tentacle to the
Pleurobrachia do this weird thing where they basically like have this super characteristic coordinated like flippy motion they do. So they have their tentacles actually pointing up all the time, and their mouth is, or no, their head part is pointing down, their tentacles are pointing up, and their mouth is up. And they do this thing where when they catch something, it goes like, they start to like rotate their whole body, and then like the tentacle kind of like slams over the mouth, and they just kind of like wipe it off into their mouth. But it's like a very specific, well-characterized series of movements, like a ballet. Anyway, just a throwback. Oh, it's not loud enough. Hold up. Redo. All right, cool. I'll tell you later. This is the species description part of the talk. So there's an amazing variety of ctenophores, and I've been talking all about like a pretty pedestrian inshore variety. But there's this huge diversity of forms in the phylum for a variety of reasons. There's ones that have no tentacles that eat exclusively other ctenophores. There's ones that have like these weird lobes. There's ones that are attached to the bottom. They don't swim around at all. There's ones that are really muscly and angry, like that one was. There's ones that specifically parasitize essentially other organisms. But the phylum is actually really pretty small, like only about 150 wink to 200 species have been officially described. And we know that there are more, but most of them live in places where they're pretty hard to get to, and you need ROVs essentially, or submersibles of some form to get at them most of the time. So we were very, very fortunate on this cruise on the Hugh R. Sharp to get a very lucky trawl. One more.
It's Vampiroctina!
Okay. So this is the ctenophore we found. So we were trawling out here off the coast of Delaware, about 160 kilometers off the coast, and very fortunate, we pulled up in our trawl a basically intact specimen of this ctenophore that maybe has been seen a bunch of times before. And we were like, this is awesome. We'll clip it. We'll get a transcriptome from it. Let's describe this shiz. So we came up with a name, Vampiroctina, for its black and red coloring. Delmarvensis in honor of the Delmarva Peninsula, everybody's favorite peninsula. So I mentioned, maybe it's been seen before, there's like this many apocryphal references to bright red ctenophores all over the world. People call them by many names, but there's no official description of any of them, and there's not even pictures for a lot of these sightings. Typically they go by like Tortugas red or this weird fake, well, not fake, not formal Latin name, Aegimarea tortugensis. So they're kind of like well known, but like as an urban legend almost. So this is just like a map of some of the sightings that are recorded everywhere from Japan to Tanzania to the Monterey Bay and a bunch on the East Coast. But it's not really clear that all of these are the same animal. But people just use that name kind of indiscriminately. So we really wanted to describe this species so we could like kind of start to bring some sanity to the whole taxonomic situation here. And in the process, we opened up a big can of worms, basically. But this is just sort of the overview of this guy's physiology. A few weird things. It's giant. It's like 8 centimeters long and like 5 centimeters wide, which is way bigger than most ctenophores. Has these bright red comb rows, which is also pretty neat. It's probably red because that's essentially black at depth. Red's the first color of light that you lose. And it has this super deeply sunken aboral organ, if you're interested. The aboral organ is like kind of the ctenophore answer to like a brain, if you want to use that as your reference point. It's basically the organizing center for its movement and other things. But it's like sunken into this really crazy deep cleft, which is also very unusual. It has a black gut, which it probably uses to obscure captured prey. And this is a joke that I've desperately been wanting to use in a slide for years. So I mentioned we opened a big can of worms, basically like while we were trying to place Vampyrotina into a family level classification, we ran into the problem that ctenophore taxonomy is a big mess. A lot of the family level classifications and above are based on like morphology from like the 19th century, and they don't really hold up to modern scrutiny. So there's all these different categories, and some of them are legit. Like Baroe is, it's like a monophyletic group. It stands up to like sequence data. But there's this whole group of order of ctenophores called Pseudipida, basically all the ctenophores that have tentacles, or not all the ctenophores that have tentacles, but a subgroup of all the ctenophores that have tentacles. It's totally split up if you look at it like from a phylogeny perspective. So yeah, it's a big mess. And so, once we got our transcriptome, sort of started analyzing the data, we got into some of these problems and started trying to come up with ways to, again, bring some reasonability and to the taxonomy here and bring everything in line with modern techniques. So we generated a transcriptome from Vampyrotina. This is me doing the most nervous dissection I've done in like a long time. We actually ended up catching like 5 of them, so it was okay. But it was touch and go. So quick cool thing, Vampyrotina actually emerges in a clade that's sister to all the other ctenophores. So it's, an early emerging lineage of ctenophores, which might make it a good thing to study in general if you want to ask questions about ctenophores more broadly, rather than looking at something that's much more derived. But the issue here is we don't actually have like a ton of taxon sampling in this analysis because it's based on whole transcriptomes. So we wanted to, better place this in sort of the general multiverse of like ctenophore taxonomy. So we used an 18S RNA approach as well, because there's been a lot more 18S sampling in ctenophores and just in general because it's easier. So there's a ton of issues that sort of emerged with this, but I'll only have time to really touch on the main one here. So it seems like Vampyrotina comes out in this group called Mertensidae, which is a family of ctenophores that's pretty well trodden. Our analysis contains like the prototypical Mertensidae member, Mertensia ovum, and it also contains another sort of like well-established member, Charastephany. But also nested within this group appears to be Euplocamus, which actually technically has its own family. And there's no sequence level justification for that act, And everything we're seeing in this analysis seems to suggest that Eubulcamidae, this monotypic family that it's in, is actually synonymous with Mertensidae. Also, weirdly, Calinera antarctica, this other broadly considered to be like Mertensid ctenophore, does not emerge in this clade. But part of the reason for that is probably because that genus is just like total garbage, basically. The original type species for the genus is actually a misidentified pteropod mollusk. And we know that, and it's been kind of corrected, sort of, but there's just like a lot of errors in the taxonomy for it. And it's just a total mess. So it seems like all of this is in need of a bunch of revisions. We're undertaking that in a description paper. But a lot of people are sort of less than enthused by that because we're trying to bring ctenophore taxonomy in line with molecular sequence techniques. And a lot of taxonomists are super old school, and they're just like, everything should be done by morphology, even if it doesn't make any sense. So we're trying to make a strong case for doing things differently, bringing things in line with modern techniques and thought. But it's been interesting going. I've learned a lot from it. But I'll be happy to talk about any of these other random issues that have come up later. And that's all I have to say for you right now. And I'm going to finish up here. Close out on a philosophical note. No one alive, whether they're ctenophore or human, is more or less primitive than anything else. We've all been evolving for the same amount of time, and we should celebrate and cherish the unique aspects of all living things' physiology. And we shouldn't think about things as being complex or primitive and stuff, because it is more likely to reflect our biases than anything else. And the true story of what's complex or primitive or whatever is likely bogus or difficult to assess, even if it's not. Yeah, and I hope you come away, having expanded your consciousness from this. So now I'm going to thank just a bunch of people, everyone in my lab. You're all lovely. Some of them are not here. Dylan is not here, and Lincoln is not here, but they're here in spirit. It's Dylan Fox. That's a joke. Okay. Anyway, but all my lab, everyone, you're great. Allison, you're awesome. My wonderful collaborators, Gabriel, who's chilling, I guess, in Puerto Rico right now. I think he's a junior now. Mike, who often makes very good decisions. Nathan, who's just a lovely person, and Alex, who did that, who's that Pokemon voiceover. All have been working with me on describing this ctenophore. Nathan in particular, I just wanted to shout out because he's done so much to teach me about taxonomy, and it's been just really interesting learning experience. We've also worked with Ken Hlanich, Mike's advisor, and Nathan's former postdoc advisor, who's also given us a lot of great input. I'd like to thank my committee, all of y'all showing up, and our funding sources, and just a ton of other people. I've gotten to travel a lot in my studies, and I've met so many awesome people, I can't even thank them. But I do want to shout out the CEB Microscopy Corps, because I would not have been able to do like half of this without them. And yeah, thank you, every one of you. And now just like a ton of sentimental stuff, thanks to my parents, who've been really amazing, and my dad, this is like super dramatic shot of him. All my friends, some of them not here, Joe in Latvia for whatever reason, Michael in Boston, the Latvia of New England, I don't know, and a few of my friends who are in the back, and everyone in BMB, y'all have been great. And also my partner Katie, who's here and who's been immensely supportive of me the past couple months, and also brought food, which hopefully we'll enjoy after this. We have a mirror cake, so fire up the gram. And yes, everyone, there's 27 people watching on YouTube, which is about, I think, the number of people in here, if not more, which is pretty cool. So thanks to chat. I haven't been looking at chat the whole time, so I have no idea what kind of crazy shit's been going down in there. And yeah, with that, I'll just take any of your questions. Whoa. Yes.
I'm really curious about the process of, you just brought up this organism from the vaccination, and who knows how long it's going to be alive, or if it's still alive. What do you do to
That's a great question. First thing is to calm the fuck down, which took a few minutes. We also benefited immensely from having like several people kind of there and sanity checking each other, because I think we were all kind of coming up with like, oh, what should we do first? All kinds of crazy shit. But so what we did, and I think this is like pretty good, we immediately got it into like a holding tank and started taking photos. We also had a few people writing down notes, just everything. Everyone was just spitballing. Anything they could see, we were recording, in word form. And people were asking questions like, oh, where's this canal? Or like, what color is this? And so just taking down every single bit of information you can, and really doing that basically as exhaustively as possible. And then after that, and only after that, thinking about, okay, let's cut this thing up. And then once you say like, let's cut this thing up, it's relatively straightforward. There's only a few places where you might want to cut it up. And then you do that. And then try to think about preserving the rest of it as best you can. Because technically to do a species description, you do need a holotype. Our holotype is preserved to the best of our abilities, which is very not high level for ctenophores because they basically are very fragile. But yeah, that's basically the process. Just write everything down as fast as you can, take as many photographs as you can, and try to keep each other like in line. Like, oh, I don't think that's red. I think that's actually like red-orange. Oh, okay, cool. It was alive. Yeah, it's, go back to that video. This is the state it came up in.
You found one.
We found 5 in the end, but the pictures I'm showing you here are just the one that was in the best shape. We killed every single one.
So you may have just killed all 5 of the last 5.
Yeah, why didn't you kill
I mean, that's like, it's like that quote, it's like the oldest pine in a tree, right?
I'm not here to demonstrate that I'm a good person, just that I'm well defended as a person. No, yeah, I mean, the thing is, you can't even really put things back when you've taken them from depth for the most part, because actually going back, people try to do that with fish sometimes, and it just like never works out. Let me go back to this video. Yeah, so this was the one that came up in best condition. So it's moving its combs around. It was actually probing around with its little tentacles. It was very active, surprisingly active. I mean, people were in disbelief when we showed them, like.
How come it doesn't blow up?
Because it doesn't have like air in it. Like fish have like swim bladders and stuff, and they have air pockets, and those expand.
Is it in salt water?
It's actually being kept in water that we collected at the same depth. Yeah.
How do you like equalize the pressure? Because you put the water into the tank, right?
Yeah.
In, I don't know, some crystal depth, and then you're moving it up. So how does the pressure correlation work in that volume?
If you have a swim bladder and you come up in our trawl, you come up not in your best condition.
But like if you don't know, I mean, even still, there's still some, like is the pressure inside that tank the same as it was?
It's leaking. It's not a perfectly airtight or watertight seal. Yeah, yeah.
And that's like slow enough that it's like adiabatic and these things are able to.
Well, it's not that slow, because like fish, like their eyes pop out. I don't know how else to say it.
But these are used to 70 atmospheres.
Yeah, but they're just like the body is basically just salt water. There's basically no air inside of it. There is actually like
Probably suffered from that.
they probably not though. Actually, the people have shown that the gel inside actually stores oxygen. So like their metabolic needs are super low, and they're carrying basically everything they need with them in like a pretty stable state inside of them. But so we've drawn this like this. Actually, it was definitely had like a little air bubble in it that kind of made a little blip there. So there can be a little bit of damage, but for the most part, there's no pockets of air or anything to push anything out, and that's why.
So that's true for all the gelada?
Let's see. There's gotta be some gelada that has like an air pocket somewhere in it.
So like when you pull up a jellyfish, it
Yeah, no. When we pull up, we also pull up like Atolla and other, actually Atolla from, whoa.
these are
Yeah, no, we didn't pull Katie from the deep sea. So we pull up Atolla all the time, and they're in perfectly good shape as well.
As far as you know, you've never seen them.
I've never asked.
You've never seen them like that. You mean at that time?
Well, so people actually have observed these things at depth, but it's an interesting thing that you point out, though, because there is a big issue with, like, people go down these ROVs, and it's dark, right? There's basically no light. So you have to shine light to get videos and stuff. And when you do that, you're basically, like, these things aren't used to there being any light, and then you are shining like these giant lamps at them, and they go blind, basically, almost always. And so, like, their behavior is not necessarily characteristic of, you know, what it might like be like otherwise. What? Totally. Cosigned. Yeah. Yeah, ctenophores are self-fertilizing hermaphrodites, probably. They also spawn, probably. There's a whole, there's a lab in Florida that's working a lot on that. Actually, yeah. So Monterey Bay Aquarium has figured out how to keep them in culture, as have a few other people. So you can keep them in culture now, and there's also a few people who are working on doing tissue culture of ctenophore cells, mostly muscle cells and nerve cells. You can spawn them. I have spawned and raised some to like a larval stage. You need to be really on your shit if you want them to grow to adulthood, but it can be done. And hopefully will be done in the coming years. But the tissue culture is actually pretty interesting. I think it's, well, if you look up a friend of ours, Laura Vanderpas, she's done a bunch of work on that with Bill Brown. If you're really interested in, you know, doing morpholinos and stuff on them, I think they're interested in that as well. I can confirm not tasty. That's all I'll say. Okay. Jeff. Thank you.
My question is about the adhesive cells.
Yes.
Are they, so is the idea that, like, when the tendril gets poked, these adhesive granules actually pop because of pressure, or is it like, is there some mechanical sensation that's making cells like secrete from the granules?
Yeah, good question. So, debatable. But, so they don't necessarily always trigger if you touch them. You can observe this just by like sort of touching the tentacle lightly with like a fork or like a glass rod or something. It's not always going to stick. Sometimes it will. There's been a bit of work done on that. There is probably some kind of mechanical trigger that certainly seems to work. I guess if you bop it hard enough, it will also just rupture because it's not exactly, made out of steel. But, so nematocysts have like a little pressure switch, basically, like it will detect a pressure wave and fire. And I don't think anyone's identified something like that in a ctenophore, but it's triggered. It's not necessarily abundantly clear quite how, but that's what, it's presumed to be kind of similar. What's up? It's a good question. So we actually haven't looked at I saved some of the tentacles in formaldehyde, but we haven't done like microscopy on them at all. Presumably, yes. But it could be if it's closely related to Euplocamus. I didn't actually say this, but Euplocamus has striated muscle, which is unique in the phylum. Nobody knows how that happened. So we haven't looked at, for instance, if this has striated muscle, but the reason that's interesting is because Euplocamus actually can kind of like really grab things with that striated muscle. It will like wrap a tentillum around and like, pull it in. So it also has colloblasts, but it's also like grabby. But yeah, it's almost certainly got colloblasts because it's present in most ctenophores of this type. We haven't actually looked technically. Thank you.
Okay, so whether you use a mini or a lesion or well,